Good morning everyone, and thank you for joining the meeting of the Telenetro project, funded by the Prima Foundation and by the national agencies of each participating country. Agencia Estatal de Investigación, AEI, State Research Agency, Spain. MER, Ministry of Universities and Research, Italy. Ministry of Higher Education, Scientific Research and Innovation, Morocco. Ministry of Higher Education and Scientific Research, Tunisia. The project is titled, New Low-Cost Crop Strategies Based on Biodiversity and Remote Sensing to Reduce the Application of Nitrogen Fertilizers in the Mediterranean Area. Its objectives are the design of a new cultivation strategy aimed at reducing the contribution of nitrogen fertilizers through the use of plant sand or botanical extracts capable of slowing down the transformation of ammonium into nitrate. Monitoring the nutritional status of crops using remote sensing and/or ion-selective electrodes. This project is developed in the context of agricultural nitrate pollution. Agricultural nitrate pollution severely affects soils, groundwater, and coastal ecosystems across Spain, Italy, Tunisia, and Morocco. These regions face eutrophication, salinization, and environmental degradation that threaten agriculture and socio-economic stability. The project brings together leading research centers to develop sustainable strategies, combining fertilization management, plant physiology, microbiology, and remote sensing to reduce nitrate contamination and improve water and soil quality in vulnerable Mediterranean areas. Today, the use of synthetic fertilizers in agriculture is still excessive. The most common nitrogen fertilizer is urea, followed by ammonium nitrate and ammonium sulfate. The problem is that these fertilizers release large amounts of nitrate into the environment and contribute significantly to nitrous oxide emissions. In this project, we aim to rely mainly on ammonium-based fertilizers to avoid adding more nitrate. However, ammonium is quickly converted into nitrate in the soil, which is a major challenge we need to address. Here you can see the nitrification process that takes place in soils. When ammonium from a fertilizer reaches the soil, nitrification begins very quickly. This process is carried out by two main groups of bacteria, Nitrosomonas and Nitrobacter. These microorganisms convert ammonium into nitrate, unless something inhibits or suppresses their activity. Once ammonium is converted into nitrate, it moves rapidly through the soil to deeper layers, where plant roots can no longer absorb it. As a result, nitrate is easily lost, and ends up contaminating natural ecosystems such as soils, groundwater, and eventually rivers and coastal waters.
One of the strategies we are applying in this project is the use of plants or botanical extracts capable of inhibiting the conversion of ammonium into nitrate. In the case of BNI plants, we have carried out a series of greenhouse studies. First, we created a seed bank of plant species that, according to the scientific literature, can release root exudates that slow the nitrification rate. Here you can see the plants that we have used in the experiments. These plants are Brachiarias, Posis, Gramineas or aromatic plants familia.
Plants were grown in a multi-tunnel greenhouse during the summer period, in 0.5 L pots filled with sieve soil, collected from the project's experimental plots, free from pathogens and weed seeds. For each of the 20 plant species, 3 nitrogen nutrition treatments were established using modified Hoagland nutrient solutions, 100% N, full-strength Hoagland, 75% N, 50% N. For each species and nitrogen treatment, 6 plants were used. The plants were grown in pots filled with soils from MRSA, and we tested them under different nitrogen levels. The nitrogen concentration of 14, 7.5 and 3 mmN applied like nitrato ammonio and unisolucin nutritive Hoagland. The plants grew under these conditions for 3 months. And then plant growth parameters were measured. Fresh weight of shoots and roots was recorded at harvest. Soil nitrification rates. Root metabolites. Chemical analyses of root tissues like non-structural carbohydrates, total soluble sugars, reducing sugars, and starch. Oxidative stress markers, hydrogen peroxide, HO, and melangialdin, MDF, antioxidant capacity. In the photo you can see some of the plants used in the experiment. In the responses of all these plant species to the nitrogen doses, 3 types of responses can be observed. One group of plants shows their maximum growth at the medium nitrogen level. This includes rice, canola, and grass. For example, in canola, when moving from the 14 mm treatment to 7.5 mm, growth increases by 51%, and when moving from 3.5 mm to 7.5 mm, growth increases by 308%. Then we have a group of plants in which the highest growth is obtained with the highest nitrogen concentration in the nutrient solution. These plants are Brachiaria hybrid, forage cabbage, Brassica oleracea var, Sudan grass, sorghum times Drummondi, maize, Z maize. For example, from N50 to N100, Brachiaria decamans increases its growth by about 133%, while Brachiaria mombasa shows an increase of about 100%, and we have a group of plants in which the lowest nitrogen concentration produced the maximum vegetative growth. This group includes millet, sorghum, green soybean, peanut, and tall fescue. For example, in green soybean, growth at 50% nitrogen was much higher than at 75% and 100%, showing a clear preference for low nitrogen availability. Another measurement we carried out was the calculation of the nitrification rate. It was performed as follows. Another measurement we carried out was the nitrification rate. To do this, we prepared soil samples by mixing 15 grams of homogenized soil with an incubation solution that contained ammonium sulfate, sodium chlorate, and a phosphate buffer. We adjusted the moisture to about 60% of field capacity and incubated the samples for 20 hours at 26 degrees Celsius in darkness. During the incubation, we collected subsamples at several time points, 1, 2, 3, 6, 8, and 20 hours, and then centrifuged them to extract the soil solution. These extracts were frozen until we measured nitrite using the grease method. Here you can see an example of how the nitrification rate is calculated. We measure the nitrate concentration at different times, and the rate corresponds to the increase in nitrate over that period. For instance, between 48 and 76 hours, nitrate increases from 5 to 9 milligrams per gram of soil. So, the nitrification rate is simply the difference divided by the time interval, in this case, 0.14 milligrams of nitrate per gram of soil per hour. In this slide, you can see nitrification rates for all the species in the experiment. To make the results easier to compare, we divided the plants into two main groups. The first group, shown in Table 1, includes the species with low nitrification. These plants show very small nitrification rates across all nitrogen levels, in many cases, almost zero. This means that these species are able to slow down or suppress the conversion of ammonium into nitrate. This group includes most Brachiaria species, the millets, sorghum, Sudan grass, tall fescue, mint, canola, and forage cabbage. The second group, shown in Table 2, includes the species with high nitrification. These plants allow a rapid transformation of ammonium into nitrate, with values similar to bare soil, especially at 100% nitrogen. This group includes rice, maize, peanut, sorghum bicolor, millet CP, and others. This is another example of how the nitrification rate varies over time and with nitrogen concentration. For green soybean, the maximum nitrification rate occurs at 6 hours, and only under the highest nitrogen level, 100%. At medium and low nitrogen, nitrification remains very low. For sorghum bicolor, the peak also appears early, around 3 hours, and again only with the 100% nitrogen treatment. The 75% and 50% levels show almost no activity. For Brachiaria mavuno, the highest nitrification rate occurs at 6 hours, but only with 100% nitrogen. Under 75% and 50%, nitrification stays close to zero throughout the measurement period. Overall, all three species show their maximum nitrification under the highest nitrogen concentration, with very limited nitrification when nitrogen availability is reduced. This slide shows an example of the biochemical analyses we performed on the roots, including phenolics, starch, soluble sugars, and MDA. When we look at phenolic compounds, we can see that each species responds differently to nitrogen availability. In green soybean, phenolic content is highest under the 100% nitrogen treatment and decreases at medium nitrogen levels, showing a partial recovery at low nitrogen. In sorghum bicolor, phenolics decrease sharply as nitrogen is reduced. indicating that this species produces fewer phenolics under nitrogen limitation. In Brachiaria mutica, the trend is the opposite. Phenolics increase as nitrogen decreases, reaching their highest value at the lowest nitrogen level. Overall, phenolic accumulation is highly species-dependent, and in some plants it increases under nitrogen limitation, while in others it decreases. This is an example of the untargeted metabolic study that we are currently analyzing. This approach allows us to identify both primary and secondary metabolites that may be involved in the inhibition of nitrification. We can see that the heat map changes, depending on the nitrogen concentration in which the plants were grown. In other words, the metabolic profile is completely different under each nitrogen level. Now I am going to show you one of the specific chromatograms. In this chromatogram, we can clearly see that the intensity of the sorghum peak changes depending on the nitrogen level. Higher nitrogen concentrations show a strong and well-defined peak, while lower nitrogen reduces its abundance. This indicates that sorghum production is nitrogen-dependent and may play a role in the plant's regulation of nitrification. The chromatogram of sorghum shows a dominant and characteristic peak that is very stable under high nitrogen levels. As nitrogen decreases, the peak becomes smaller or shifts, suggesting that sorghum synthesis is sensitive to nitrogen availability. Since sorghum is a known BNI compound, these changes may explain differences in nitrification inhibition among treatments. In this figure, the Brachiaria tone signal varies dramatically with the nitrogen supply. Under optimal nitrogen, the chromatogram shows a sharp and intense peak, while under nitrogen deficiency the profile changes completely. This demonstrates that Brachiaria tone production is tightly linked to nitrogen status, and may be a key metabolite driving BNI capacity in Brachiaria species. Now we are going to go through a few brief comments on the botanical extracts. We have a collection of 45 botanical extracts provided by several companies. The company that contributed the most to this collection was Atlantica Agricola, and another company donated the synthetic nitrification inhibitors that are commonly used in controlled-release nitrogen fertilizers. The study to determine the nitrification rate was carried out by the private company Idagro. Nitrosomonas europaea was cultured under controlled conditions, and then exposed to each plant extract at a fixed concentration. The mixtures were incubated, and samples were taken every hour to monitor nitrite production. We added DCD to immediately stop the reaction, and the nitrite formed was quantified using the grease reagent. This allowed us to determine how strongly each extract inhibited the nitrification activity of the bacteria. This slide shows the group of extracts with the strongest nitrification inhibition. As expected, the synthetic inhibitors DMPP and IMSA appear among the top performers, showing the highest negative nitrification rates. Interestingly, several botanical extracts also showed very strong inhibitory activity, such as equistum, thymol, citrus blend, lavandin, eucalyptus, and citronellal. These results indicate that some natural extracts can reach inhibition levels comparable to commercial synthetic inhibitors.
The final and general conclusions are: several plant species showed strong BNI capacity, especially Brachiaria, millets, sorghum and aromatic species, consistently reducing soil nitrification. Nitrogen availability strongly modifies plant metabolism, affecting the production of key BNI-related compounds such as sorghum, brachiariactone and sorghum. Multiple botanical extracts showed strong nitrification inhibition, and several natural products performed similarly to commercial synthetic inhibitors. The project has identified promising biological tools, plants and extracts, to reduce nitrate pollution and support low-cost, sustainable fertilization strategy. These results provide a solid foundation for field validation and future integration with remote sensing technologies. We are currently analyzing the chromatograms from the untargeted metabolomic study, which will help identify additional metabolites involved in BNI activity. Thank you very much for your attention.